So, hello everyone again and welcome to my channel. Really excited to be back again. I actually talk with you all about something quite interesting related to, I'll say, biochemistry. Specifically, we'll be looking at enzymes, and we talked a little bit about various enzymes and how they are actually involved in catalyzing various biochemical reactions in the body. And one thing that comes to our minds is as follows: how do we classify all these different enzymes that are involved in various reactions? One way is to be able specifically to follow a specific guideline under the Commission classification, which uses the EC numbering system. And specifically, we're going to look at the 6 classifications of enzymes based on the reactions and based on their systematic names, and also an application of this naming system for those enzymes with respect to the substrates and products that are actually produced from the introduction of the enzyme into a specific reaction. So in here we have the EC name, and the EC, which is the enzyme class, has all the 6 numbers right there. So we have number 1, which is our main focus, and in this case we're going to look at number 1 classification, which is called the oxidoreductase or oxidoreductase. So that means that this particular type of enzyme, you notice it's a protein with the ase at the end. This is actually involved in redox reaction, which means it undergoes oxidation and reduction reaction between 2 species, either in terms of the transfer of hydrogen atoms or the transfer of oxygen atoms, and specifically transfer of electrons from one molecule to another. And a systematic name to approach this type of enzymes is first putting the oxidized species name first, and then you write down the reduced species name, and followed by the final name, which is the general name oxidoreductase. Now you can also write another way. There's another way of writing this. This is just writing the substrate name and then put down the hydrogenase as the general name. So an application of these is, first, let's look at the top one and look for a name to actually apply to this particular format. So one is actually called alcohol reacting with oxygen. So in this case, alcohol will be our oxidized species, and then oxygen will be our reduced species. And what is going to happen is that there will be an oxidoreduction, which in this case is going to be written as oxidoreductase. And if you want to write it also in terms of just the substrate, the hydrogenase, you can just pick the alcohol because it's more significant in this case, and we just write the hydrogenase. Exactly. So now we're done with the first class. We're going to move to the second one, which in this case is transferase. And transferase involves the transfer of one functional group from one molecule to another. So once you're able to remember that for EC2, we can then apply what we know from the oxidoreductase into the transferase naming system. And in this case, you have to focus first on the one that does the donation, which is called the donor, and then you look for the acceptor in that specific reaction, and then finally you add the group name that is involved in the transfer, which from one to the other, and then finally we actually put down the general name, which is transferase. Or we can go with a simple one which we don't really go into, so I'll just ignore that because this is pretty much the generic name for transferase in general. So in this case, if we go to an application of this, we actually have an amino acid called D-alanine, and this is the donor, and we have our acceptor, which is 2-oxy or oxo. Just go back. Where's reader? Yep, oxoglutarate, which is the acceptor, and the group that is transferred from the D-alanine to the 2-oxoglutarate is actually called the amino group. And then you put your final name, which is transferase. Yeah, so that is it for the second one. And now moving on to the third one, which is hydrolase. So in this case, we need to remember that since it's hydro, we know that water is involved in here, and it involves in the breakage of a bond. So in this case, it's called the hydrolytic cleavage in here, where water is actually used to break a specific bond. So in this case, we actually figure out that to follow the systematic naming, we actually go with the substrate name first, followed by the group name, and then the generic name, which is the hydrolase. And in this case also, we can actually go with another simpler name, which is just putting down the substratase. So substrate is kind of the prefix, and the suffix is ase at the end. So an application of this is following a specific compound, which in this case we call a Acyl glycerol, sorry, and this will actually have the group which is actually going to be cleaved, which is called the acyl group, that it contains it doesn't have an R, so I'm going to take that away. Yep, there it is. And this is going to perform the hydrolysis, where water comes in, hydrolase, where water comes in, sorry, and cleaves this particular bond that contains the acyl group right there. So another way of naming this is by actually using just the name that we know, which is a nitrile. This can be another enzyme in here. We can just do a Hydrolase. That will be another form of actually applying that to this particular example if you want to. And then the next one is a Lyase, which is actually the enzyme, I think the enzymes class 4, and this one is actually involved in the elimination reaction that forms double bonds, or the addition reaction of a double bond. So in this case, we can actually go from triple bond to a double bond, or you can actually add stuff in to a triple bond in order for you to reduce it to a double bond, or vice versa. Just the creation of a double bond is actually facilitated in this particular process. So in this part, what we need to know is that for the systematic name, we actually go now with, let me try and get this right, we have the substrate with the eliminated group, and then you put Lyase. And so in this case, we have to put the substrate name first, followed by the eliminating, or I would say the eliminated group in this case. And then finally, we put an hyphen and Lyase at the end. Or you can just go with the product name, and then at the end you put synthase. There is a little bit of a mix-up somewhere when we get into the last one or so, and you have to be aware of that. It's actually important in this particular exercise right here. So an application of this is L as parate, and this as aspirate is going to have the eliminating group to be ammonia, and as a result it performs a lysis in a way, or I would say the lyase is an enzyme that does the elimination of ammonia from the L asparate compound right there. So in this case we actually form our product, which is called asparagase in a way. But anyways, that is just going ahead of myself. So moving into number 5, which is EC5, we have isomerase, which means is something going on within a specific compound. This is actually a form of an intermolecular rearrangement in a way, where bonds are actually like rotated or actually broken within a specific chiral center if there is one, and then there is a switch in orientation in a way in the 3D space, and causes that particular compound to be different from a rhizomer or a telomer. So there are various names to actually apply in here. Like for example, you can actually call your substrate to actually be either going into a specific form of an isomer, which is called a racemic structure, but this is caused by a race maze. Another one is epimerase. Another enzyme can be substrate isomerase. And another enzyme in here can be, I would say, substrate. I'll just kind of force it in here. I'll drag this up. Telomerase. So an application of this one right here for the substrate telomerase is, for example, we can have a ketone telomerase, cetomerase, or we can have one in here with the substrate racemase. And an application of this one right here is, let's say, an alanine, which is a type of protein, or sorry, amino acid, and this will have a racemic mixture or a racemic structure. There we go. And then finally, ligase, that is the one that we were talking about, where the bond formed is coupled with ATP hydrolase. So in this case, take note that the product first is the name, and then you're going to put in the family name, which is synthase, not synthase. So in the lyase, we actually went to synthase, But in this case for ligase, we actually go with synthase because ATP is involved in this hydrolysis process. And so you can also do that, or you can go with another way, which is writing this first substrate and then the second substrate, and most importantly putting down the final name hyphen Lyase. So an application of the one at the bottom here is as follows. We actually have an amino acid L-aspartate, I'll put the T here, let me erase this, get that out of the way. Aspartate, ammonia, and in this case we'll put a hyphen and ligase, or on the other side the product that is formed from this particular ligase influence in this particular reaction is the formation of the product, which is called the L-asparagine synthesis. Yeah, so that's about it for this particular video. Hope you're able to understand all the 6 classes of enzymes and how the reaction are actually performed, and also the reaction how it links with the systematic name, and finally an application of applying the systematic names in there. So all the same, don't forget to hit the like button, share and subscribe, and let me hear your thoughts about this. By that way I can talk to you later. Stay smart and believe in yourselves.